Welcome to Ritomo's Foam Tool demonstration. This is Ritomo's user interface, where we have loaded the CT data of a scanned foam. First of all, we need to perform the automatic segmentation process. In this case, there are two materials present, the cell and foam materials. We can safely assume that the cell material is air. We adjust the threshold, while trying to accurately enclose the wall material within the yellow lines. After that, we run the seeds and grow functions to obtain the final part image, where a material has been assigned to each voxel. This is the model and volume view, after disabling the slice's visibility. This way, we can easily visually examine it. We can now start preparing to use the foam tool. It is recommended to enable the slice's visibility and switch to the no volume view. The foam tool is used on the part image, which can be found in the item browser. We need to select the cell and wall parts and adjust the marker size. Let's run the detection with the default settings. As we can see, several coalesced cells seem to be classified as one big cell. That is not the analysis desired outcome, therefore we need to modify the settings accordingly. When further separation of the cells is needed, a smaller marker size should be used. By adjusting it to 1 and rerunning the detection, we achieve a better result. The tool's results are stored in the foam data item, where they can be accessed again later. There are various visibility options. Let's deselect the Draw Walls option and keep only the draw cells. Some neighboring cells might appear to have remarkably similar colors. By enabling the Show IDs option for the Z-axis, we can determine whether it is one or separate cells. For example, this area might appear as one large cell, but upon further observation, we can conclude that it is in fact two cells of the exact same color. We can also only enable the Draw Walls option, including an approximate thickness legend. We activate the Draw Cells feature again. By clicking the Cells List button, the Foam Cells Details window appears, allowing us to view the geometric characteristics of each cell. We can draw the cell principal axes, which derive from each cell's most compact and elongated directions. It is also possible to export the table contents and the cell voxelized meshes. This is what they look like in ANSA. This is the Walls Details window, where the approximate wall thickness distribution graph is displayed. The vertical zoom switch can be toggled to enhance visual inspection. By checking this box, the assumed wall midline is drawn in red, from which the wall mid-surface meshes are created. Moreover, we can export the wall thickness histogram and smoothed or voxelized mid-surface meshes, including the thickness information. This is the smoothed wall mid-surface mesh in ANSA. And this is the voxelized wall mid-surface mesh. These are the two meshes side by side, and by activating the element thickness graph we can confirm that the thickness information from Ritomo is preserved in these meshes. Thank you for watching.